we see that most of the times the observations are independent, so the values on one observation, they do not tell about the values of the other observation. But sometimes the observations may be dependent on one another. So we do have some dependencies within our data. And these dependencies may arise due to different reasons. For example, if you look into the within array, there might be some duplicate spots which are located next to each other. So since these are same spots but duplicated, so they are kind of correlated with one another. So you might want to take their means or the averages for these two spots, and maybe you want to combine these spots with one another. Sometimes you have same sample across multiple slides. So in this way there is a correlation. So the sample is the same. So you might want to keep track of this information. You might have samples from the same patients or subject. So in this case, the samples obtained from the same subject or the same patient, they are correlated with one another. How to deal with these dependencies? We can use the models and that account for correlations, since those observations are correlated with one another. So we can fit in those correlations into our models. We can combine those dependent observations into single observation. We can take average observation from replicated spots. We can pair up the data. So sometimes you have different conditions. They are the measures on the same subject, or otherwise if they are measures on different subjects, you can use unpaired data analysis. So here is the pictorial representation. So here you see only one distribution, and each individual data point represents the difference between the two data points which are kind of paired or kind of coming from the individuals who are maybe correlated with one another. In order to do a t-test, it's just like doing a one sample t-test. So we have this distribution of these differences, and you can come up with the mean of this distribution, and then you can plug into the mean with this t equation, which is, as I said earlier, it's the actually equation for one sample t-test. So we see that data points are here, they are somehow correlated, and we have to pair up the data points with one another, and we can calculate their differences, and we can test our hypothesis using the distribution that represent the differences between different data points. So again, we consider this TP53 example in patients before and after a treatment with zinc chloride. So we are using the same individuals here. So there is patient number 1 before treatment and after treatment. So this patient number 1 is then different than patient number 2, and then this is different than patient number 3. But for the same patient, you can calculate the individual differences before and after treatment. So in this way, the variability between different patients can be taken into account. We want to test whether the drug is affected or not. In order to this drug to be effective, we want to see higher TP53 expression. So after treatment, P53 protein should be more in these cells. Same way, before this P53 protein is there, since we are doing the expression studies, their messenger RNAs should be high. So since we are capturing messenger RNAs, we are kind of making our hypothesis that the gene expression is higher. So since these samples are correlated, we have to run a pair t-test. We can compute the differences in expressions for each pair of genes. We define those differences as treatment versus control. For patient number 1, the treatment has, the number is 15, minus 12, the number is 3. Same way we can calculate for rest of them. So in this way we can come up with a column of differences. We can take this column of differences as a distribution of differences, and we forget about the individual data points over here. So next our tests are going into, we'll use only these D's. When we come up with their mean, it becomes D bar, and we can throw one sample t-test on this. So in order to do that one sample t-test, we do those hypotheses where we assume under the null hypothesis that there is no difference, or the difference is less than 0. So in that case, the control has a difference, and the control minus the treatment, the number is a negative number, or this number is equal to 0. Whereas our alternative hypothesis is that difference is greater than 0. So if it is big number, This number should be big enough that it should be present into the tail or the right tail of this distribution. Our calculated difference is 2.4, the mean difference. When we come up with standard deviation, it was 0.89, and we can calculate the standard error. So that is SD bar. So that is 0.40. We plug in into that equation, we come up with T calculated, that is equal to 6. Since we have 5 differences, our degrees of freedom is equal to 4. So null hypothesis or the null curve is having a degrees of freedom is equal to 4. So when we draw that curve with degrees of freedom is equal to 4, we plug in different values. Our critical T at 0.95, our P is equal to 0.95 or 95% up to that point and 5% towards the right, that number should be 2.13. So any calculated number, T calculated greater than T.13 is a significant event. In terms of difference, this number should be greater than 0.85. So our calculated number was 6, which lies somewhere here, and probability up to that point is 0.998. If you compare with alpha, this region has a probability of 0.019. So this is 1 out of 1000. The probability of finding this event by chance is 1 out of 1000. Our T calculated is greater than T critical, and our probability of this T calculated is less than alpha or less than 0.05. So it's a significant event. So it's the same data which we used earlier. The values are same, but doing it as a pair t-test, we were here, since we fit in the right model, we can come up with the significant results. So there are some drawbacks or disadvantages of combining this data. So sometimes we lose information and it does not account for the original variability, as well as we assume that all combinations are equally effective.